Cell transport processes can be broadly broken down into passive or active, depending on whether ATP or energy is used. Passive processes involve simple diffusion, osmosis, and facilitated diffusion, and involve the movement of particles down a concentration gradient. Simple diffusion involves the movement of particles such as oxygen and carbon dioxide that are small and usually nonpolar that can fit directly through the cell membrane and go down the concentration gradient. Osmosis also involves the movement of particles down a concentration gradient, but specifically water or solvent molecules. Facilitated diffusion is also the movement of particles down a concentration gradient. However, it involves the movement of these particles using some sort of carrier or transport protein. Things that are too big to squeeze directly through the cell membrane, such as glucose, need some sort of protein to push them across. Active transport refers to those that involve the use of ATP. There's two subcategories of active transport, pump and vesicle. Pump includes things like the sodium-potassium ATPase pump, which moves sodium out and potassium in using ATP as a source. Vesicular or vesicle active transport involves two subcategories, exocytosis and endocytosis. Exocytosis involves a vesicle docking at the cell membrane and releasing a particle into the extracellular environment, while endocytosis involves the opposite. It involves the engulfment of some sort of particle into a vesicle that is now in the cell. Endocytosis has three subcategories: pinocytosis, phagocytosis, and receptor-mediated cytosis. Pinocytosis involves the engulfment of liquid particles. Phagocytosis involves the engulfment of foreign particles like pathogens by white blood cells, while receptor-mediated endocytosis is a very specific form involving the activation of receptors. The specifics of these aren't as important for Module 1, but keep them in mind. Follow to stay on top of prelim bio.